Okay, welcome everybody to this Validate Early Career Researcher Showcase. We have two excellent speakers today. My name is Blakely Nixon. I'm the Comms and Events Coordinator at Validate. Today we are going to hear from two early career researchers from two different continents actually today. Our first speaker, who I'm going to introduce now, is Dr. Chandni Yaqoob, who is an independent researcher based in Pakistan and has been educated at the University of Punjab. And today I'm going to hand over to her now. She's going to be talking about fusion molecules from Mycobacteria tuberculosis antigens. So over to you, Chandni.
Hello everyone.
And I can hear you perfectly.
Okay. Good morning, everyone. My name is Chandni Yaqoob, and first of all, thank you so much for giving me this opportunity. I am very thankful for Validate Network, and let me share my presentation screen first of all. Okay, so is it visible to everyone?
Yep.
All right. So I will be presenting 1 minute, and am I also visible? Okay, is everything all right?
We can't see you, but it's okay. We can see someone called admin, but don't worry. But we can see the screen. We can see the slide.
We can also see Dr. Chandni's webcam.
Okay. So I will be presenting work from a laboratory at the School of Biological Sciences, University of Punjab. So my topic of research was designing fusion molecules from antigens of Mycobacterium tuberculosis to enhance serodiagnostic sensitivity in latent TB infection and active TB state. So what was the problem? To understand why this research matters, consider the scale of the problem. As of 2025, tuberculosis caused 1.2 million deaths and produced 10 million new cases annually. So more alarming is the latent TB infection, latent TB reservoir. An estimated 2 billion people worldwide carry the infection silently, as all of you know. So that is without symptoms, representing an enormous risk of future active disease. So as you know, the available diagnostic tools each have critical shortcomings, as spear microscopy is very sensitive, and culture test, although it is more sensitive, but it requires weeks of incubation. And moreover, IGRA is quite expensive, and it is restricted by cost and operational complexity. And TST is although cheap, but it gives less sensitive results. So, and as you know, while GeneXpert revolutionized TB diagnosis through molecular precision, so the future of serodiagnosis lies in antigen innovation and combinational diagnostics, not in replacing, but complementing the molecular tools. So coming towards the objective, our study had a focused 4-part objective structure. First, we used in silico tools, specifically BaPyPred-1 and BaPyPred-2, to locate and to predict and validate B-cell epitopes in our chosen antigens. Second, we constructed fusion antigens. So we engineered 2 fusion molecules, and that is RB2C26C with RB2031C and RB2C26C with truncated RB1827, and we expressed them in soluble form in E. coli. And the third step was the structural characterization. So we confirmed the correct protein folding via CD spectroscopy and We validate the epitope accessibility by doing molecular modeling by using different softwares. And the last thing is, and very important thing was the evaluation and validation of those fusion constructs by testing ELISA sensitivity and specificity on plasma from total 300 people, including 116 ATP patients, 90 latent TB individuals, and 94 healthy cohorts. So the underlying hypothesis is that presenting multiple immunogenic epitopes simultaneously in a single fusion construct will increase the diagnostic sensitivity without compromising the specificity. So what did we know going into this study? 3 antigens had established immunogenic credentials. So these are the 3 antigens which we chose for our study. That is RB2C26C. That is also called a hypoxic response protein 1. It's a DOSA regular antigen, upregulated under low oxygen, a key condition during latent infection, and has been shown to generate both humoral as well as cellular response. And the next one is the HSPX, RB2031C, that can induce responses in both latent and active TB. It is also an antigen from the DOSA regulon, and it is upregulated during the latent phase of the infection. And the third one is the RB1827, that is also called GAR-A, and it had been previously reported to show outstanding sensitivity and specificity in active TB plasma. So, however, the literature had some significant gaps. So no study has systematically combined these specific antigens as fusion molecules and tested them against a rigorously confirmed TB cohort using quantiferon gold as the reference standard. And while a single antigen sensitivity were reported, they rarely exceeded 35 to 40%, far below clinical acceptability. So we need some reliable and low-cost and cheap diagnostic method to diagnose so many people in a TB endemic country. So coming towards the methodology. So our experimental workflow was organized across 5 integrated stages. We begin computationally predicting B cell epitope positions using Bepipred 1 and 2 with a threshold cutoff 5, selected to balance peptide length and location accuracy. And moreover, another software, that is IEDB database, another database was used, additionally used for RB2031C epitope data. So we collected data of that HSPX protein, HSPX antigen from IEDB. Then we cloned the 3 individual genes and engineered 2 fusion constructs. RB2C26C fused in frame with the full length RB2031C and RB2C26C fused with truncated version of RB1827, from which we truncated 61 amino acids and from the N terminus in order to improve the solubility. So all constructs were expressed in E. coli with N-terminal histachs and purified by nickel affinity chromatography with greater than 87% purity. So the purity, and that is confirmed by SDS page and also by doing densitometry analysis. So for structure validation, CT spectroscopy was performed using a ChirScan plus spectrophotometer, and 3D models were generated via RaptorX and 3D Refine. Next, ELISA was performed Before going to ELISA, let's quickly go through the cloning and expression results. Due to shortage of time, I want to show you the results quickly. Let me show you another results. So we begin by isolating the genomic DNA isolation, genomic DNA. So we used the strain H37RV genomic DNA, and from there we amplified. From this, we amplified 3 native antigens by doing PCR. So as you can see, these bands. Next, we confirm the presence of these inserts by doing restriction analysis of these native antigens, and then coming towards the protein expression. So this shows the expression of all these 3 native antigens, these bands. So the first one is for RV1827, next for RV2C26C, and the third is for RV2031C. So then we did a protein purification of all these native antigens. That is for RV2C26C, and then we purified all these proteins by doing the nickel affinity chromatography. So you can see all these purified bands, and then we got the purity more than 90%. So there was some problem with this RV2C26C that after purification, it showed dimer formation. So I removed its dimer by heating this protein, by treating with 150 millimolar DTT at different various temperature. So at high temperature, the dimer removed, and then I again purified it and made it dimer free. So this is the schematic designing of my fusion construct. As you can see, this black box represents the epitopes. So first we join the RV2626C with the RV2031C, and then we made another construct by joining the RV2C26C with the truncated version of RV1827. So that's how we cloned our fusion. So we use PET28A for expression, and then we confirm the presence of inserts of fusion construct insert by using the restriction analysis. And then again, we go through the same procedure, the protein expression of our fusion antigens, protein purification, and also we did Western blot by raising the antisera, polyclonal antisera in rabbits. Now, coming towards the key results of ELISA. So after doing that gene cloning expression and purification, so ELISA was performed on 116 active TB patients from we did sampling from the Gulab Devi Chest Hospital, 90 latent TB individuals confirmed by quantiferon TB Gold, and 94 BCG vaccinated healthy controls. And we did statistical analysis by using the Crystal-Wallis test and ROC curve analysis by using graph pad prism 9. So coming towards our core results. So the core finding is presented here both in table form as well as in graphical form. So first let me explain this. So among the three native antigens, antigens, sensitivity for active TB ranged narrowly between 30 and 34%, insufficient for clinical use. For latent TB, RV1827 performed particularly poorly addressed 2.2%. So the fusion molecule changed this picture dramatically, and RV2C26C, RV2031C fusion achieved 50.9% sensitivity for in case of ATB, and 63.3% sensitivity was achieved in case of LTB. So both fusion construct maintained 100% specificity against the BCG vaccinated healthy control group. So this is the scatter dot plot that makes the magnitude of improvement visually clear. So you can see that plot showing the antibody response of single and fusion antigens. And these colors, the blue color indicates healthy control, red ATB, and the green color indicate LTB high individuals. Okay. So beyond that, beyond sensitivity and specificity, we used ROC curve analysis to assess the overall diagnostic accuracy. For the latent TB group, RV2C26C, RV2031C showed the highest AUC of 0.1 in excellent discriminating capacity. And for active TB, RV1827 performed best at AUC 0.87, followed closely by RV2C26C, truncated RV187 at 0.86. So next, we did protein characterization by doing the CT spectroscopy as well. So CT spectroscopy showed that the secondary structure profiles of alpha helices and beta sheets match closely with the predictions from both Morphold and RaptorX software, confirming the correct folding. And next, we also did the protein modeling. So these are the structures of native proteins, which some of these structures we taken from the different websites like Swiss model or PDB, and some of the models we generated. And we then visualized all these models by using Discovery Studio, through which B-cell epitopes in yellow color and solvent accessibility for antigen-antibody interaction shown in the blue color. So coming towards the main molecular modeling of this fusion proteins. So these are the 3D model which are constructed through RaptorX software, showing the B-cell epitopes in the red color. And then we did solvent accessibility analysis for antibody interaction, and that is indicated by showing the blue color. So most of the B-cell epitopes, they are solvent accessible. That ensures the antibody, and that shows the epitope and antibody binding in a good way. So coming towards the discussion, that what we interpret through our research. So the discussion indicates 5 key interpretive points. First, the fusion molecules inherently outperform single antigens because TB immunology is heterogeneous. Different patients, they show antibody response. They show variable antibody responses to different epitopic combinations. and depending on bacillary load, strain, and disease phase. So fusion molecules broadens the detection net. So second, we say that we interpret that the outstanding performance of RV26626C with RV2031C for latent TB is mechanistically explained. So as you know, both antigens that belong to the dose are regulon, that is upregulated during bacterial dormancy. So which is specifically upregulated during latent infection. So the fusion molecule is essentially tuned to the biology of the dormant state. So an AUC of 0.91 indicates strong discriminative ability. So what explains the sensitivity close to combined theoretical values? So that is the third agreement, that is between experimental sensitivity and calculated combined sensitivity. For example, 63.3 was our experimental sensitivity, and 66.7 was our theoretical sensitivity. So if we compare both these, so that confirms that epitope exposure in the fusion was not compromised by the joining. So this is the critical design validation. The fourth one was that we interpret that the discrepancy with the previously published study of RV1827 reported the very high and outstanding sensitivity of 1.9%, while our research indicated only 33.6% sensitivity. So maybe it likely due to the strain differences, and it highlights the importance of strain-specific protein characterization. Different MTB strain produce different antigens with different amino reactivities. Maybe that's because of that, because they used H37RA as a strain, as a the bacterial strain, MTB strain. They used H37RA. So we use H37RV. So maybe that will be the difference that we get lower sensitivity. So finally, and perhaps most importantly, overfusion constructs successfully discriminate BCG vaccinated healthy individuals from both latent TB and ATB cases. So let me explain what was the novelty and scientific contribution, what this study advances. So let me highlight what is generally novel here. So this is the first study to test RV2C26C plus RV2031C fusion molecule specifically against a cohort of latent TB individuals confirmed by QFT, quantiferon gold. That's the current reference standard. And the second novelty was the 63.3% sensitivity for latent TB represents some meaningful advance toward clinically applicability in serodiagnosis. There's a threshold that single antigen studies have rarely approached. So although there were some, however, there were some limitations as well. So we approached the limitations very honestly. A sensitivity ceiling of 50 to 63%, it's still very low. While a significant advance, but still does not meet WHO's threshold requirement for a standalone diagnostic tool. And the study is geographically confined to one endemic city, that is Lahore. And the RV87 discrepancy with earlier literature remains unexplained and warrants follow-up. So it needs to be, it needs to study more. So, however, the path forward is clear. The most immediate next step is to extend the fusion platform to include 3 or more antigens candidates like, for example, PSTS1, ESAT6, or AG85, which are known to contribute high sensitivity in different patient subgroups. So testing across multiple geographic cohorts will also clarify the generalizability of these findings, and given that the fusion constructs also contain T-cell epitopes. So they deserve evaluation as subunit vaccine candidates, a potential application beyond the diagnostics. So that was that's all for this study. So thank you very much. Any questions and discussions? Most welcome.
Thank you, Chandni. Does anybody have any questions? As I said, we prefer you to come on camera. I'd recommend stop sharing, Chandni, so we don't see your desktop. Just in case any messages.
Let me set my
lots of applause in the chat feature. So does anybody have any questions? Come on camera or type in the inbox or raise your hand. Any questions for Chandni about that presentation?
Can you see me, Blackley?
Hold on a sec. Go ahead. T, our next speaker. Do you want to? Go ahead.
I was wondering, Chandni, thank you for the nice presentation. It was also visually very pleasant to look at. Very nice data. I was wondering if you checked if your epitopes of the antigens, if you checked how conserved they are amongst different species, because you also addressed that there is some variance in different TB strains. Do you know how conserved these B cell epitopes are?
Yes, we checked that, and we also found that there are some non-TB bacteria, non-TB Mycobacterium species. So that's another. That will be another research. We also have to check those strains as well by using all these constructs. You're right.
And then another question I had was, because I'm not in the TB field, so what I know is that the quantiferon test, that works quite well diagnostically. So what are maybe I missed this in the beginning, but what are the advantages of using a serodiagnosis for diagnosis?
Yes, it's a very good question. As you know, the quantiferon test, it's quite expensive in our TB endemic country. In Pakistan, it's quite expensive. For one test, it costs a lot of money. So serodiagnostic test, if we can develop the serodiagnostic test by using different fusion molecules, IMIT fusion by using 2 antigens, but if we extend this to more antigen, 4 or 5 even, so it will be a better and cheaper and more reliable method for, I will not say more reliable, but I will say a cheaper method to quickly diagnose the LTB individuals.
Great, thank you.
Thank you, T, for the question. Suraj, I'll come to you in a second. We've just got one question typed from Isaac. Isaac says, you use quantiferon gold plus to classify your patients. Could you have confirmed the active cases with GeneXpert, and could that have affected classification or response pattern in the ELISA, E-L-I-S-A?
So you're talking about GeneXpert?
Yes. Yeah. It's in the chat. You use quantiferon gold plus to classify your patients. Could you have confirmed the active cases with GeneXpert?
Yes, we can. Yes, GeneXpert is a very sensitive method of detecting active TB. It directly detects the bacteria molecularly. So although it is also not much expensive. You're right. So, and WHO does not recommend current serological tests, but you know, the research is evolving, especially with antigen designs. And as I also mentioned before, that GeneXpert revolutionized the TB diagnosis through molecular precision. The future of serodiagnosis lies in antigen innovation. So we are concerning about the serodiagnosis detects the immune response, that it detects the antibody response that is generated by antigens in patients, and GeneXpert it just detects the Mycobacterium, and it is sensitive. But we also want to evolve the serodiagnostic test by using the panels of different potential, by using potential antigens, by fusing different and potential antigens and make them a better potential fusion construct. The future of serodiagnosis lies in antigen innovation and combinational diagnostic, not in replacing but complementing the molecular tools. Especially in, as you know, that in case of pediatric TB, it is very difficult to take the sputum collection. It is very difficult to collect, and also in case of extrapulmonary TB. So serodiagnosis is a blood-based test. So our research is to focus by taking sample that is from the blood, not from the sputum. In case of Gene Expert, we used sputum as a sample. And serodiagnosis focus on blood sampling. So it will be very beneficial if we want to diagnose the pediatric TB, extrapulmonary TB as well, and also it is very useful in rapid mass screening in low resource areas. So that is why we did research on serodiagnostics.
Okay, we're a bit short for time because we kind of need to move on to T now. But there are a couple of questions in the chat, Chandni. Maybe you could get back to them by just typing in the chat. But I think just for our fairness, we should probably move on to T. So T, have you got your presentation ready? But yes, Suraj, I know you also had a question, so maybe type that in the chat for Chandni as well.
Okay. All right.
Is that okay? Thank you very much, and a round of applause for using the for Chandni. Really, really good talk, and thank you for the questions as well.
Thank you so much, Blake.
Thank you. Okay, T, I'm going to start your slides, and then I will introduce you very briefly. Okay, everybody. Dr. T. Tao Pan is our next speaker. T is a postdoctoral research fellow in clinical immunology unit at the Institute of Tropical Medicine in Antwerp, that's ITM. T investigates host pathogen interactions in both cutaneous and visceral leishmaniasis. Her project methods range from field-friendly techniques and multi-omic approaches. I'm sure everybody in this chat other than me knows exactly what that means, but I don't. And today T is going to be talking about how we should rethink how we study cutaneous leishmaniasis in Ethiopia. So over to you, T.
Thank you. Thank you for the nice introduction. Just double checking I'm not on mute. All right. So I'm delighted to have this opportunity to share more on our recent publication, which is actually an opinion piece, and we discuss on how to best perform clinical immunological research in Ethiopian cutaneous leishmaniasis. So some of the things might also be quite useful for other cutaneous leishmaniasis research, not specifically in the Ethiopian context, if we have the same problems. So just to start with some backgrounds, as the audience might not all be leishmaniacs. So cutaneous leishmaniasis, CL, occurs all over the world. In fact, there are around 1 million new cases globally each year, with endemic areas spread over the Americas, South India, East Africa, and the Middle East. And this skin-neglected tropical disease is very heterogeneous, very diverse, likely due to different factors. So you have different leishmania species, you have different ethnic backgrounds of the patients, you can have a co-infection with the leishmania RNA virus, and then you also have different vectors and different reservoirs. And this can all lead to a diverse list of clinical presentations, all cutaneous leishmaniasis, but all different clinical presentations. So because this cutaneous leishmaniasis is so complex, the World Health Organization has indicated that the three main forms of leishmaniasis are cutaneous leishmaniasis, CL, mucocutaneous leishmaniasis, MCL, and visceral leishmaniasis, VL. Meanwhile, in Ethiopia, the national guidelines have mentioned that CL in Ethiopia can be subcategorized in localized CL, also mucocutaneous leishmaniasis here as a subcategory, and then diffuse cutaneous leishmaniasis. So yeah, it is indeed strange that MCL here is then indicated as a subcategory of LCL. So the conundrum starts here a little bit already. In these Ethiopian national guidelines, localized cutaneous leishmaniasis, so LCL, is present when the location of the often self-healing lesion coincides with the sandfly bite. Meanwhile, MCL is present when there is involvement of the mucosa, either through a direct bite of the vector on mucosal areas or through progression from the skin to the mucosa without spontaneous healing. DCL is present when there is a chronic disease with multiple papular or nodular or plaque lesions spread over large areas of the body. So some of the leishmaniacs in the audience might already think, this definition for MCL is different than what we have. And indeed, that's true. We will elaborate on this later. So this actually further perpetuates this conundrum. So the conundrum continues. When I started working on CL 5 years ago, we started the spatial CL study. And in this study, we wanted to investigate the host-parasite interactions in Ethiopian CL. And we thought naively back then, this will be just a relatively easy comparison by comparing LCL, MCL, and DCL with each other. But after 5 years of recruitment in one site, we saw that actually there was a quite diverse spectrum of clinical presentations. And these were actually very hard to just place in the LCL, MCL, DCL category. So before we wanted to continue with any of our analyses, we just wanted to make sure what the confounding factors were, where did this LCL, MCL, DCL come from? So we looked first at the challenges that we were going to encounter before doing the analysis. So a first observation that we had was, as mentioned before, the diverse heterogeneity in clinical presentations. And collaborators of us, they recently published this paper where they found within a group of CL experts, Ethiopian dermatologists, that in these LCL, MCL, and DCL were categories that were most used in clinically diagnosing Ethiopian patients with CL, but that this was not uniformly applied. So this means that there were issues mainly with the MCL and DCL group. So mucocutaneous MCL and mucosal leishmaniasis and diffuse DCL and disseminated CL, these two were quite interchangeably used by the physicians and dermatologists, or actually even used as a separate subcategory. So there was no consensus between these dermatologists. And another subcategory that was added was Leishmania recidivans as well by some dermatologists. So this indicates that there was inconsistent diagnosis amongst, I think they did an interview with 26 experts. So this categorizing and clinical diagnosing in LCL, MCL, DCL was not unanimous. Another suggestion that we had from other collaborators was using the term tegumentary CL. This tegumentary CL is defined as CL where the lesion involves the body's external covering. This also means that lesions that are mainly internal mucosal or when there are nodules without actually creating a lesion, just like a little bump, and other lesions that are just causing subcutaneous swelling without really actually developing a lesion in CL are not included in this umbrella term. And while this umbrella term, tegumentary CL, is used more often in the Americas, when looking it up in the WHO and the Pan-American Health Organization guidelines on leishmaniasis, it was not included in their documentation. So it is not officially a terminology that is used in this documentation. The PAHO and WHO guidelines, how they define MCL and ML, is different how the Ethiopian guidelines define it. So their definitions include a hematogenous metastases after an initial and distant skin lesion. So what's quite often there is that there's first a skin lesion somewhere, for example, on the arm, it disappears. And then there is another lesion occurring quite often in the mucosal areas, for example, in the mouth. Another definition is the extension of a facial skin lesion to mucosal membranes, and then also the direct bite of the vector on the mucosal membranes. A quite distinct representation of MCL and ML in the Americas is also the destruction of tissue. So quite often these patients have destruction of the oronasal cavities, upper respiratory tract, and the larynx, causing severe disfigurements for the patients. Meanwhile, in Ethiopia, these definitions, they're kind of same, but different. And also what's very significant in Ethiopian MCL lesions is that there is no destruction of these oronasal cavities, larynx, or respiratory tract. So the mucosal lesions are quite restricted to the boundaries of the mucosal areas. So for the Ethiopian guidelines to take over some of these definitions, but not fully, have caused some confusion in the currently available data and the interpretations of it. Then, so this has indeed led us on the currently available data on Ethiopian CL. When involving these classifications, it's difficult to make any conclusions because the diagnosis is so inconsistent amongst experts even. So we recommend actually to step away from this simplified and quite biased LCL, MCL, and DCL classification, and perhaps use a more unbiased approach. And for this, we have developed an extensive table. So in this table, we elaborate on different clinical parameters to record in future clinical immunological studies on Ethiopian cutaneous leishmaniasis. It's quite elaborate, but it's elaborate in order to make a better clinical classification later. So this table elaborates on leishmania infection history, so number of previous CL lesions, etc. Also comorbidities that might have an effect. There's an elaborate part on lesion assessments on baseline, so the number of lesions all the way to lesion morphology, where you can indicate primary morphological categories, secondary morphological categories, clinical features, mucosal involvement, etc. And then it ends with the treatment outcome, percentage of reepithelialization, for example, scar morphology, and also the details of CL treatment. So using this table, we can eventually run it perhaps even through a language learning model or an AI pipeline to obtain more unbiased clinical categories that are specific to the Ethiopian context. Another challenge that we bumped into was the assumed immunopathogenesis paradigm for CL. I think a lot of the leishmaniacs know this paradigm. So this is a figure from a very nice review made by Philip Scott and Fernanda Novaes in Nature Reviews Immunology. And what they found was that actually these three subcategories for CL, mucosal leishmaniasis, self-healing cutaneous leishmaniasis, and diffuse cutaneous leishmaniasis, were actually driven by specific immune responses. So this mucosal leishmaniasis, where there's destruction of the oronasal cavities, is actually driven by an excessive Th1 type response that causes cytotoxicity and destroys the tissue, while the self-healing CL has more moderate Th1 response, still strong, but moderately, that can control the infection and eventually resolve the infection and the lesion. And the other side of the spectrum, you have an anergic or even absent Th1 response, which then leads to a high parasitemia, leading to different lesions spread all over the body, as seen in diffuse CL. So this is a really nice paper, but for placing this in the Ethiopian context is difficult. Firstly, because, as mentioned before, these categories are not as easy to place in the Ethiopian context. Then the data that they found of the paradigm they've made is based on extensive clinical data and in vivo models, but they were restricted on infections with Leishmania major, Brasiliensis, Amazonensis, and Mexicana, so mainly South American or American species. And data for Leishmania aethiopica was not included, with Leishmania aethiopica actually being the main infecting strain for Ethiopian cutaneous leishmaniasis. Then there is some data available already for Ethiopian CL on clinical immunological research. So there's some data on the humoral response, plasma cells and antibodies, Leishmania-specific antibodies, and T-cell mediated responses. However, a lot of data is lacking on monocytes, macrophages, NK cells, and neutrophils. From this available data, when we delved in it, we saw that there were some limitations. So there was quite a lot of LCL, MCL, DCL comparisons made, which we know now is difficult for interpretation, as this is really dependent on the dermatologist that has clinically diagnosed these patients. Then the PBMCs. So the PBMCs are mainly used to define the immune response. So this is more a circulating immune cell subset, while when you're looking at cutaneous leishmaniasis, the site of infection will more be in the lesion. So lesional cells would be most informative to investigate. Then these PBMCs were often ex vivo stimulated with soluble Leishmania antigen to investigate the Leishmania-specific T-cell response or Leishmania-specific response, which is a quite good setup to do. But SLA itself also comes with a caveat. SLA is soluble Leishmania antigen. This is quite often a culture of parasites that is then freeze-thawed to create an antigen. But this SLA can come from different parasite species cultures, can be made from the promastigodes or the amastigodes stage, and should actually be defined in the materials and methods, because also with these papers, there has been some inconsistent results. I know there are some projects now to make laboratory grade and GMP level SLA to minimize this variability. There were some studies that did look at the lesion, so the site of infection. These were mainly immunohistochemistry or immunofluorescence papers. And what they did find is they did find the cells, like B cells, T cells and such, so they detected cells. But what was lacking is quite often the quantification, which allows you to do some statistics. So it will be a semi-quantification because it's difficult based on image, but this would be recommended for future studies to include always, if possible, a semi-quantification. A third challenge that we bumped into was the parasite diversity within Ethiopian CL. So as mentioned, I'm not sure if I mentioned this before, but we have actually different Leishmania species in Ethiopia. So the earliest study of one of the earliest study was in 1969, where they assumed patients to be infected with Leishmania aethiopica, region dependent, of course. And then after, over the years, they did do species typing. It was indeed Leishmania aethiopica. But after a time, it was just assumed that it was always Leishmania aethiopica up until 2006, and actually in 2023 as well, Leishmania tropica and Leishmania donovani were found from cutaneous leishmaniasis lesions in Ethiopia. So Leishmania aethiopica is not the only player in the field anymore. Another effect or another part of this parasite diversity is that more in-depth studies on Leishmania aethiopica strains have found that actually there's a lot of genetic mutation within the Leishmania aethiopica strain, with some hybrids present of Leishmania aethiopica with Leishmania donovani and Leishmania aethiopica with Leishmania tropica. So this is also an important player in the parasite diversity. And then lastly, you have Leishmania RNA virus. So this is an RNA virus that infects the Leishmania. And one study has found that in their Leishmania aethiopica parasite isolates, half of them were actually co-infected with Leishmania RNA virus. Whether or not this co-infection can lead to a more virulent strain of Leishmania species still needs to be investigated. So also here in these studies, there were some limitations. So again, there was this one study where they compared the genetic variability of the Leishmania aethiopica from different patients and compared LCL, MCL, and DCL groups. They had some additional groups as well, and they found no difference. But this, as mentioned before, comes with a caveat, this comparison. Then there are different sensitivities in the species typing. With species typing, I mean that you then look whether it's Leishmania aethiopica, tropica, or donovani. There are different methods available. But important here is that when you want to look at hybrids, for example, a lot of these old school methods don't work anymore. So then it's better to look at maybe biobanking your sample appropriately and look for experts in the field that can run it in their lab. Some of the papers also characterize the parasites based on a culture. So this means that they've taken a sample, they culture it in the lab to have more yield of parasite to then characterize them. And while this is a good method when your yield is not sufficient, you are creating a bias by only characterizing the faster growing parasite strains and not the slow growing ones. So I think recently now in the past years, there have been some more methods present where you can directly characterize your parasite species and even in-depth analysis directly from the clinical sample. So now, having identified these three main challenges, we have created some key considerations to address in our future clinical immunological research, but also for others, of course. And a first thing that we want to emphasize is to taking on a multidimensional approach. So when you're starting a study, to already think of perhaps performing clinical, immunological, and parasitological assessment in parallel. And this can be done with appropriate sampling. So you could take then lesion-derived material for both your immunological and parasitological assessments. And important to note is that when you're taking lesion-derived material, like this is quite often a lesion biopsy, is to indicate where in the lesion that you will take it. Because, for example, if you take it from an active border, it will not be as comparable as maybe a more anergic middle area of the lesion. So these are things to also indicate. Then a second consideration is to actively use public databases and maximize your biobank inventory. regarding clinical data sharing, for example. So if you want to use the data of the patient and also biobank samples for secondary use or collaborations later after your project ended, this should already be specified when you're writing the clinical study protocol. And if we want to really follow, well, you should really follow the ethical guidelines, there should be a tick box in your informed consent form where the patient can indicate that they allow secondary use of their biobank samples in clinical data. And then you will have to specify if the secondary use is then focused only on Leishmania or also other pathogens. Because, for example, you might have co-infected patients with a helminth infection, and if you want to do a collaboration with an helminth group later and share their samples or data, it should be indicated on the ICF. What is also really nice and helpful is registering your clinical study at clinicaltrials.gov, even though your study will not be an intensive trial and might just be an observational clinical study. It is nice to already register it, not only for sharing your study protocol and your assessments with the field, but also because, yeah, clinical studies, they take a long time. And by the time you have a publication, we are a couple of years further. So it's nice to refer to this registry, also when grant writing, for example, while you're waiting for your publications. On your publications and also at the clinicaltrials.gov, you have the opportunity to provide details for accessing the data. So this allows other researchers that might not have sufficient samples or do not have the funds to start a new clinical study to perhaps use your details and collect them for a metadata analysis, for example, to reach out in an appropriate way and request data. So for us in the Institute of Tropical Medicine, we have a data access committee. So often institutions have their own department for it, and they usually have an application form to access the data. So for sequencing data, a lot of journals now obligate the authors anyway to do this, is to deposit your data in public repositories like the European Nucleotide Archive. And this is really nice because then your data is then on a public server and then can be used to be run or included with different cohorts or different studies as comparisons. And it can, if needed, also be rerun with optimized computational tools. Because even though you have the data, we only have so much limited time and also so many resources. It's good to have other experts as well to rerun it, especially with such complex data. Then regarding your sample biobank, after every clinical study, there are always leftover samples or samples that have been reserved for later analysis. So when you're starting your clinical study, it's already very important to think of keeping your samples long-term for later collaborations, perhaps. And with appropriate storage, I also mean that your sample is already dissolved in the correct buffer or not even in a buffer. And usually, if it's frozen already at -80 degrees or -60 is also okay for long-term storage. This will allow your samples to be processed or even reprocessed, so a bit similar as the previous part, with optimized and advanced methods. We are living in a very fastly accelerating world where I feel every year there's a new advanced method. So by even waiting with processing your samples 1 year, I'm sure there are new advanced methods in more high throughput way that can allow you to run your samples in an even better manner. Something that I found is actually lacking in the Leishmania fields, and they started doing this for TB already, is to have an open access platform, a site really, where there is a reference for all the available sample biobanks from different research groups all over the world linked to published studies. So this is nice to actually start new collaborations and to include more cohorts in your study for doing nice comparisons. Then a last key consideration is local investments. And we wanted to address this because there are all these suggestions and these recommendations, but to keep it sustainable, it's important to do huge investments in the local research infrastructure. So what is often done in international collaborations is that there's quite often a donation of the instruments after collaborative projects. So in Ethiopia, where we work, we could buy a new flow cytometer or a centrifuge or all the pipettes, and then these are donated after the project has ended. Whenever we start a project as well, we also perform recurrent trainings beside the site initiation visits, like all the methods are re-explained again, even more simple ones like an ELISA or the PBMC isolation. And we actually have to do this as well because what we've noticed in the past is that there is an immigration of specialists. So you're training a very nice good group of researchers, but of course, everybody gets opportunities abroad. So when there is immigration of these specialists, the expertise is also lost in the local infrastructure. So we've noticed this in the past, I think 5 years. So we're now trying to focus more on training the trainers. It's a bit of a more heavy investment, but it has already shown some nice results. Like a nice example is the Leishmaniasis Research and Treatment Center at the University of Gondar. So in 2004, I forgot to place this here, but the center was built in 2004 by DNDi. And then in the past 10 years, DNDi has led their projects over the last 24 years there now. And in the last 10 years, ITM has also been allowed to collaborate with the team. And with this collective efforts, this is a group of 20 clinical and laboratory staff that has been trained and retrained every time on these projects. And they've also remained, making the team only stronger and experts in their field. And in 2022, this opened then more opportunities for them. So the University of Gondar then provided the opportunity to build an extension lab, an extended clinical trial laboratory that's following the GCLP and GLP rules. And now in 2025, this has been finalized, and this clinical trial lab is actually now also open for other clinical projects, so not Leishmania restricted, and will also be taken up by the Ethiopian network of clinical trial laboratories. So this is a really nice example of heavy local research infrastructure investment, but that has led to really nice results. Another nice example is a bioinformatics capacity strengthening training that we have done with our institute and ARI in the capital. This was done 2 years ago, where some of the ITM members then trained a large group of Ethiopian bioinformaticians in bioinformatical analysis of pathogens, so very pathogen focused. And in the second module, a selection of these trainees have then become trainers to then train a next group of bioinformaticians locally. So we hope to continue this and just continue to train the trainers so that this expertise remains locally. A lot of recommendations and suggestions have been mentioned, so I just want to keep the takeaways concise. Ethiopian CL is very complex, as you have seen, and still under investigated. So the main takeaways are: make your clinical studies future-proof already from the beginning, writing the study protocol and the ICF as elaborate as possible to protect yourself later as much as possible. It's important to remain unbiased and identify confounding factors with the recommendations provided. In the American CL, there's a lot of research that has been done, but to just take over the assumptions that they have found to the Ethiopian context has not been really appropriate. And then, yeah, since we are working on a skin-neglected tropical disease, the funding landscape is quite limited compared to oncology, for example. So sharing really is caring. Like we have to work together to accelerate research on this under-investigated and very complex disease. So all the suggestions and recommendations can be found in this very recently published opinion piece. And lastly, I would like to thank the Spatial CL Consortium. This is the first paper to come out, and soon, yeah, this year will be a big paper publication year for the rest of the Spatial CL papers. Thank you.
IT, I'm going to put that. I'm going to put that in the chat. The article, and then so thank you very much. Lots of rounds of applause in the with the responses. Do we have any questions for Dr. Pam? Do we have any questions? And again, you can come on camera, you can raise your hand, or you can type in the chat, and I can try and. Suraj, do you have a question? You want to come on camera?
Thanks, Blakely. Sorry, my camera is not working, unfortunately. Good talk, T. Just want to check with you. Your focus was mostly on CL, and then you said in Ethiopia, last major parasite is Donovani, which is not in CL parasite. So can you comment on that?
Indeed, it's a quite interesting and recent finding. So Donovani is more an infecting strain associated with visceral leishmaniasis, but we actually found it as well to develop cutaneous leishmaniasis lesions in some of the patients. And we're thinking maybe this could be, there's still, it's still being investigated more in depth what this strain looks like compared to the visceral Leishmania donovani strain. Maybe this is it's a bit similar to, I think, what is found in Sri Lanka. You also have a Leishmania donovani there that's causing CL. So it could be a sub-Leishmania donovani strain that creates cutaneous lesions as well.
Oh, okay. Is there any chance that these patients are infected with more than one species of Leishmania?
If they were, it should be present in the species typing. We are collaborating with Professor Jean-Claude Dujardin. He's the parasite specialist in it, and he's doing more of a deep dive on what strains were found. But I remember from that specific study, it was very clear there was only this Leishmania donovani. And there's now an ongoing study to do more in-depth sequencing analysis on these strains.
And one last one. Is there a possibility of that Leishmania RNA virus changing the virulence of CL or Donovani to go from one spectrum to another spectrum?
That's a good comment. I don't know that much of Leishmania RNA virus, but it could be possible, and it could be something that they can investigate as well when they have the sequencing data. So I'll share that comment. Thank you for that.
Thank you. Good job.
Hi there. We have a question from Jonathan. What would you advise for sampling from a lesion based on your experience? One, be consistent in taking samples from either active border or middle portions of the lesion, or two, take samples from both active border and the middle portion, clearly indicating the specific site. It might be better if you just read it in the chat.
Yes, I'm reading it. So yeah, in an ideal world, you would take a sampling from the border and the center to just have two very significant different places, but you will also have to consider what is first ethically allowed from the IRBs. So when you're taking a 4 millimeter biopsy or a 2 millimeter biopsy, that already makes a difference. Because if you take, for example, 2 4-millimeter biopsies, these are quite large. That means that your lesions will also have to be quite large as well. And then you're indirectly also selecting for patients with larger lesions. So it depends on your research question as well. I think, but then again, you also want to have sufficient sampling. Sufficient large sample for your analysis. I know for immunology, if we want to look at the cells from a 4-millimeter lesion biopsy, we get around 1 million cells, of which the majority are leukocytes. But if you just want to look at the parasite, for example, then you can take 2-millimeter biopsies, which would be more likely easy to take from the border and the middle. And then a second consideration to do is to also explain to the patients, because biopsies are quite scary sometimes, what the consequences are. So what you could tell the patient as well is, and what from our experience we found is when you take a lesion biopsy, even 4-millimeter in diameter from an active lesion, it heals completely because your lesions, once they're being treated, they're in the healing process, so there's no scarring.
Thank you. We have a question from Veronica. Does the diversity of clinical presentations impact treatment decisions?
Yes. So at the moment, according to the national guidelines, if a patient is diagnosed with MCL or DCL, they immediately get systemic treatments. So it's quite more invasive. While if they are diagnosed with LCL, they get more local treatment like cryotherapy or an intralesional injection. But then again, there are some exceptions if this LCL.
What are the second years? Proses ideal. Normal market.
Oh, sorry. Jonathan? Jonathan, can I ask you to mute? There we go. Sorry about that, T.
No worries. So it does impact treatment decisions. So that's why that's why it would be good to have a better clinical classification system also to optimize treatment, because now there are some guidelines, but also in practice, physicians follow their gut feeling more as well.
Oh, great. So we have a question from your fellow speaker, Chandni, who asks, if we move from blood lesion-based studies, how might that change our understanding of the disease?
I wouldn't necessarily move from blood to lesion. I would add the lesion focus. So it's actually quite nice to have both, because when you're looking at the site of infection, especially with Leishmania aethiopica, cutaneous leishmaniasis, sometimes it's considered more of a local infection, but when you see cases like diffuse CL or like when there's more lesions all over spread of the body, it's likely moving through the circulation as well. So it's good to have the added feature of circulating cells.
And I think final question to wrap up, Kieran asks, what lesion-specific immunology signatures or pathways do you think should be prioritized to develop a more accurate immunopathological model for the disease?
I would not want to look at a certain pathway necessarily, because then you are already putting yourself in a kind of bias. Like generally in Leishmania, we already know that the Th1 response is generally the response that is protective. But then if you already focus on this T-cell response, perhaps you're also missing important other responses that have not been investigated yet. So I would just stay as unbiased as possible until you actually have the first data of your research question. So for example, with our spatial CL data, what we will do now is we will use computational models to then look at in which clusters or which areas there is more upregulation and more differences, and then look in each pathway separately, if it makes sense or not, and how big the differences are. And then based on the data, you focus then on a specific pathway.
And I'm going to be a bit cheeky and maybe sneak one more question in from Toraya in Tunisia. Did you analyze the lesion microbiome to check any potential co-infection?
We have not in the spatial cell, but there is actually another group in our institute that started such a study in Ethiopia. So I'm not sure if that study is already on clinicaltrials.gov. I can look for it and then send it to you, Turaya. But what they are looking at is they're checking the microbiome, as you mentioned, on lesions, and then what they do is they will do a kind of clinical trial as well to have an antibiotic topical treatment to see if that improves the treatment as well. Because Ethiopian CL is also known to be difficult to treat.
I think that's right. As a follow-up, what about the intralesional immune response with the different CL?
So currently we are investigating that. So the analyses are ongoing, and I hope to have this submitted by the end of this year. So hopefully that paper will come out next year. So it will be an analysis of the lesions of Ethiopian CL using single cell and spatial transcriptomics methods.
Okay, that's great. I would highly recommend that both T and Chandni just have a scroll through the chat, because there's lots of very nice people saying how good the presentations were. So I would just like to thank both of our speakers, Chandni and T, for 2 excellent talks. And I would also like to thank everybody for coming and for some brilliant questions and a rather busy session chat, which I think is just a testament to how engaging these talks were. So thank you very much. I'm going to stop the recording now, but thank you very much for coming, and we'll see you in the next one.